So in continuing with immunity, we already know that there are two main types of immunity, acquired and developed. Acquired means which you acquire during the lifetime of an individual, and develop also means which you develop it during the when you get sensitized to a pathogen. So similarly, when we talk about immunity, we already know that there are two types of immunity. One is cell mediated, another is humoral mediated immunity. Both these types of immunity is due to lymphocytes. What they do is the lymphocytes are produced in the bone marrow. These cells, one migrates to the thymus gland, and once it reaches the thymus gland, it matures producing T cells. The other remains in the bone marrow itself and produces B cells. B cells are the ones which produce antibodies. So today we will be discussing about the structure of antibodies. Antibodies is also known as humoral mediated immunity. In addition to this antibodies, B cells also produce cells called memory cells, which also help in producing antibodies, so that when the same pathogen infects an organism in the future, it requires a rather shorter time for the production of antibodies. So when we talk about antibodies, it basically consists of two chains. These are the heavy chains and these are the light chains. Both these chains are bound with the help of disulfide bonds. Now, at the ends of both the heavy and the light chains, you have a variable difference of amino acid sequences. So these are known as the V regions. V means variable, whereas the remaining portion of the light chain and the heavy chain are known as C regions, which means constant sequence of amino acids. In addition to this, the heavy chains each consist of 5 different types. They are known as alpha, delta, epsilon, micron, etc. So depending on these, the immunoglobulins or antibodies are known as IgA, IgD, IgE, IgM and IgG. G stands for gamma. So your antibodies are also known as immunoglobulins. So there are 5 types of antibodies or immunoglobulins. There is IgA, IgD, IgE, IgM and IgG. Why is it called such? Is these alphabets represent a constant sequence of amino acids which they might represent. That is, these heavy chains might be an alpha. If it is alpha, then the immunoglobulin or antibody is called IgA. If it is delta, it is called IgD or D. So when we talk about antibodies, the most abundant antibody or the primary antibody involved in fighting infections is IgG. About 70% of the human serum contains antibodies, that is IgG. And the most less is IgD, whereas IgE is concerned with allergies, and IgA is passed through the breast milk. So when we talk about antibodies, this is the structure. Now how the antibodies basically act on pathogens is, I've already told you, it is made up of 2 chains, heavy and the light chain. This gap between the heavy and the light chain is the site for binding antigens. Antigens are basically proteins produced by the pathogens. So So once it binds to the antigens, what happens is there are three ways of action. One is agglutination. By agglutination, what it means is the antibodies bind to the antigen and they form huge complexes which are easily removed by the cells. Second one is opsonization. By opsonization, again what it means is the antigens bind to the antibodies. As such, when they bind to the antigens, they make the antigen or the pathogen susceptible to phagocytosis. And the last one is cell mediated lysis. By this, what it means is, again, it renders the antigen of the pathogen incompetent, and in turn it invites other cells to act on the ineffective pathogen. So this is how an antibody works. So I already told you that our IgE antibody is basically concerned with allergies. So how allergies work is due to a massive release of histamines by the mast cell. How histamines work is they work on two receptors, H1 receptor and H2 receptor. H2 receptor is found in the stomach and it is basically concerned with the release of gastric juices, whereas H1 it is found in the skin, lungs and the gastrointestinal tract. So what happens is when histamines are released abundantly, H1 receptors they become active. As a result, the most common symptoms that you observe during allergies are swelling, redness of the area, itching, and in some very rare cases, the allergies might also lead to severe conditions. Such severe condition is called anaphylactic shock. So this is a condition where the allergy becomes very severe and in some cases it might even lead to death. When we are talking about immunity, it can either be of two types, natural or artificial. Natural, it can again be of two types, innate, that means the immunity that you are born with, or acquired, which you develop during the lifetime of an organism. Artificial means immunity produced artificially, and one of the main components of providing immunity artificially is vaccines. So basically when we talk about vaccines, what it does is we are injecting a weak organism, that is a pathogen, or products of the pathogen into the body of a host, so that the host produces antibodies, and these antibodies remain in circulation of the host organism, so that when the same pathogen infects the organism, these antibodies can easily fight against it. So vaccination is an artificial means of immunity. So when we talk about vaccines, they are generally of three types: killed, attenuated, or toxoids. By killed vaccines, what it means is this vaccine basically contains organisms which are dead or killed, but still these dead or killed organisms are sufficient to sensitize the immune systems. Attenuated means injecting the individual with weaker strains of the pathogen. And finally, toxoids means the harmful proteins or antigens produced by the pathogens. So generally when we talk about vaccines, every country follows their own vaccination routine. Like when you were kids, you were all given polio vaccines up to a certain age. Then also there are vaccines for tuberculosis, and so on. Polar and now, interferons are substances which are produced by a cell infected with the virus. And once interferons are produced, they provide immunity to the remaining cells from the virus. So interferons, they are not already produced, but rather they get produced only once the cells are infected by the virus. So interferons are a very important group of compounds which help in providing immunity against viruses. So when we talk about immunity, there are also some certain huge cases like autoimmune disorders. Autoimmune disorders is what it means is basically the immune cells are supposed to recognize a foreign particle. But in autoimmune disorders, what happens is the immune cells act on self cells. That means they act on cells of the individual itself. As a result, they might produce diseases like Hashimoto's disease. In Hashimoto's disease, what happens is the immune cells start attacking the thyroid cells, leading to abnormal thyroid conditions. Basically, immune cells are supposed to fight against foreign particles, but in this case, in autoimmune disorders, what happens is immune cells act against the body's own cells. So, when we talk about immune disorders, there are various examples like SCID. SCID stands for Severe Combined Immunodeficiency. Basically, in this condition, what happens is B and T cells are not produced. As a result, the individual cannot fight against even simple infections. Such individuals which have SCID are kept in germ-free environments. Another one is AIDS. Again, it stands for acquired immunodeficiency syndrome. It is basically caused by a virus, and in this case again the immune cells become very weak. They do not fire off against the diseases. And another immune disorder is George's syndrome. Again, in this case the B cells are not functional. So these are the immune disorders. So, in order to overcome immune disorders, there are a wide variety of practices, especially in the biotechnology field, and one such thing is recombinant DNA technology. So, when we talk about recombinant DNA technology, it is also used to produce antibodies. We all know that there are 5 types of antibodies, and not all 5 are necessary to fight against a particular antigen. So what they do is, we require only one antibody, which is sufficient. So production of only one antibody is known as monoclonal antibody. So how monoclonal antibodies are produced is, we infect a mice with a particular pathogen. Once the mice has been infected with a particular pathogen, it will produce antibodies only against that particular pathogen. So we select the spleen cells and inject it with a myeloma cells. By myeloma cells, what it means is myeloma cells are cells which have cancer. Basically, these are tumor cells. So, spleen cells plus myeloma cells produce a hybrid cell called hybridoma cells. These hybridoma cells are grown in culture. So the spleen cells are responsible for the production of antibodies. The myeloma cells are for the increase of the cells, because these are nothing but cancerous cells. So once they are grown in culture, we get huge amount of antibodies, and these antibodies are used to fight off against infections. Another way of also providing immunity is by using already preformed antibodies in other organisms. Like for example, when we are treating a snake bite condition, so in order to produce antibodies against snake bite, what they do is they already inject a horse with the venom. So the horse produces antibodies, and these antibodies are circulated in the body of the horse. Again, they are taken out from the horse and injected into a person who is suffering from snake bites. So this is how immunity is